In this video, we will discuss about a new strategy, that is RIBOMAP, in order to profile whether a particular mRNA is translating within the cells or not. This can be useful to check whether there is a down regulation or up regulation of particular mRNA during specific condition or specific kind of stress. So let us understand in brief how this strategy works. This strategy is usually use three probes. That's why it's called triprobes. So suppose this is the normal condition in which the mRNA is translated, and this dark blue is or this blue color or cyan color represented the ribosome that is bound to the mRNA. Or this is the zoom in figure. This one is the non-translating mRNA, and this one is the translating mRNA, in which the translation is under process, or we can say co-translation is going on of the mRNA. So this probe will detect what are the mRNAs that are on. This probe will detect whether the mRNA of our interest will present in this phase or not. If it is present in this phase, then it is the possibility is very high that this mRNA is translating. So how these processes work? First step is the hybridization of this splint probe to the 18S RNA site of the ribosome. So this way the splint probe is hybridized, and this what happens along with this is the known as padlock probe. This padlock probe is bind to the specific site of interest of mRNA. So suppose this is the specific site of interest of mRNA, and here this padlock probe is bind. And this third probe is known as the primer probe. So what it will does is it will helps in the DNA amplicon. And what happens if this mRNA is present but it is not translating? Then the padlock probe will be there and the primer probe will be there, but there is no splint probe. And because of the absence of splint probe, the ligation of this padlock probe will not be happen, as this ligation is supported. This ligation of this padlock probe is supported by this splint probe. After that, and this ligation step is shown in the next step. In this case, what happens is that after ligation, this padlock probe will become circularized, and this primer probe primer probe will help in initiation of the initiation of the amplification of this DNA, and which is called here as DNA amplicon. Whereas what happens in this case? In this case, the linear padlock probe is there. Because of this, the amplification of this probe will not be possible, because there is a breakage that occurs whenever the translation of or amplification of this DNA is being attempted. And this thing is shown here, the rolling circle and amplification. And as this thing, what happens is that this DNA amplicon will rotate in this direction, and this mRNA will translate it in this direction. This will lead to the formation of this DNA amplicon, which is shown here. And this padlock probe will contain a specific kind of barcode that can be used to detect whether this mRNA of a particular interest is translating or not. So at this moment, what happens is that some of the DNA amplicon is present, for which the mRNA translation has been observed within the sample. After that, this DNA amplicon will then be transferred to this this hydrogel tissue chemistry, from which hydrogel tissue chemistry and SIDAL sequencing then helps in reading out those signals that are present in the DNA amplicon. And by decoding these signals and and annotating these signals and referring to the design, we can finally predict which DNA amplicons are which DNA amplicons are present within the samples, and which mRNA are upregulated or downregulated, or which mRNA are translated within the samples. So, and this is the legend that is shown here, and, and this panel B represents their different kind of control experiment. So in case where everything happens correctly, all triprobes are uh, present close to each other, then they will be able to see the signals as it is shown here. And in case there is no primer probe, they will not be able to see any signal. This is considered as a negative control, and this is also the negative control, in which without any splint probe they will not see any kind of signal. And if this we can say splint probe does not contain or contain incomplete hybridization part, then it will not bind to the ribosome. In this case also, we did not see uh, any visible signal for the DNA amplicon, and all these things are quantified in the panel C, where we can see that triprobe signals are quite high, whereas other signals, that is without primer probe, without splint probe, and incomplete splint probe, the signals are significantly lower compared to the signals of triprobe. So this further substantiate their technology, that is RiboMap, to uh, identifying whether a particular mRNA is translating within the sample or not. And it can be compared with the wild type samples to understand whether it is upregulating or downregulating. If you need to understand this technology, or understand about this uh, RiboMap profiling in details, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.